Down for a number of questions from each chapter. It's it's basically just whatever PowerPoint's the biggest or whatever chapter's the biggest, we'll have more questions from, and then it just kind of tapers off from there. But anyway, so 12 was the biggest, and then there's still a sizable amount from these other chapters. You guys are used to the format by now. Most of the questions will be multiple choice, but then there'll be some true false thrown in. Make sure you bring a number 2 pencil, all that jazz. Any questions about like the format or anything?
Are you gonna make those questions that you had like 3 answers to?
A, B, C.
Those are my least favorite.
Yeah.
No.
Yeah, no.
There might be 1 or 2 of those again. Yeah, there's not. I mean, they're not a ton, but there could be 1 or 2. They could raise their ugly head. And by could, I mean will.
Well,
I think. I honestly, I don't remember. I'd have to go look at it again, but I think there might be 1 or 2. No more than like 1 or 2. Yeah. Any other general questions?
Which one do we have to spend 30 seconds on?
What's that?
You always say with one of the tests you gotta spend 30 seconds on one thing.
Yeah.
36.
36.
36.
Okay,
Yeah, it was a little
36.
too much. Yeah, that's right. All right, so let me go through the content here. I'll ask you questions. You can ask me questions. So we start with chapter 12. It's a big chapter. What are the 2 main divisions of your nervous system? Central and peripheral, right? What's in the central?
Brain and spine.
Brain and spinal cord, and then what's in the peripheral?
Everything else.
Basically everything else. The nerves, the special sense organs, all that stuff, enteric plexuses, et cetera, et cetera. Now there are 3 subdivisions of the peripheral. What are the 3 subdivisions?
Parasympathetic. No.
Somatic, autonomic, and enteric, right? Those are the 3 divisions of the peripheral nervous system. Which one of those 3 is voluntary? Somatic, right? Remember, autonomic and enteric are involuntary, and that's because the motor part of somatic always goes to skeletal muscle, whereas the motor part of those other guys, they either go to smooth muscle, cardiac muscle, or glands, right? Remember, by definition, somatic sensory neurons are carrying signals toward your central nervous system, and if it's somatic sensory, these are sensations that you're consciously aware of. So touch, pressure, pain, itch, tickle, that kind of stuff. Somatic motor neurons, they always carry signals away from the central nervous system, and they always bring it out to skeletal muscle because you can voluntarily control it. For the autonomic nervous system, autonomic sensory are connected to more internal receptors, like monitoring your blood pressure, monitoring your blood glucose levels, things like that. They're things that you're not consciously aware of, right? It's subconscious. And then for the motor function is involuntary because it goes to smooth muscle, cardiac muscle, glands. And the motor function also has 2 subdivisions. What are the 2 subdivisions of the motor part of the autonomic? Sympathetic and parasympathetic. What's the nickname for the sympathetic? Fight or flight. What's parasympathetic? Rest and digest. Good. All right. Hold on. Functions of the nervous system. 3 basic functions: sensory function, which is carried by sensory neurons; integrative function, which is like analysis, etc. That would be like the interneurons that are inside your brain. And then the last function is the motor function, and that's carried by the motor neurons. In terms of the histology of nervous tissue, what are the 2 cell types you find in nervous tissue? Neurons and neuroglia, right? Neurons get all the attention because they send the electrical signals, but the neuroglia are still super important. Neurons, what are the 3 main parts of a neuron? Cell body, dendrites, and axon. Good. The cell body part has most of the regular organelles that you'd find in any other body cell. There was a special kind of clumping together of rough ER. What's that called? Missile bodies, right? That was something special you could see. All right, moving right along here. Now, when I drew my neurons, do I have to redraw this? Nope. I don't even have to redraw it because I wrote it already. All right. What's that shape of a neuron called? You were close. Not multicellular. That's called.
Unicellular?
What is it?
Unicellular?
No. Multipolar. Right. That's a multipolar neuron. All motor neurons are multipolar. Also, most of the neurons in your central nervous system are multipolar. That's the most common type. What was this type called here? That's unipolar. Most of your sensory neurons are like unipolar. And then what was this type here? That's bipolar. That's the least common type. a lot of your special senses, like inside your nose, in your retina of your eye, they have like those bipolar neurons. All right, so those are the three possible shapes. Now, the neuroglia, they don't get the love that the neurons get, but they're still important. Which do you have more of, neuroglia or neurons?
Neuroglia.
Yeah, by as many as what? 25 to 1, right? So neuroglia can outnumber neurons 25 to 1. Some specific neuroglia, what's the names of the neuroglia that participate in phagocytosis? Let me look it up. It's the microglia. They do the phagocytosis. So basically they're there to be they're kind of like part of your immune system. They're there on the lookout for bacteria, viruses that might get into your brain tissue, and they'll try to gobble it up, help defend the system, right? What's the name of the neuroglia that make myelin in the peripheral nervous system? Schwann cells. Who makes myelin in the central nervous system? Oligodendrocytes. Who makes the CSF? Ependymal cells. We'll talk more about that later. And then who are the guys that make the blood-brain barrier? Astrocytes. By the way, what does the prefix astro mean? Anyone know? It means star, and cyte means cell. So these are star cells. They kind of look like little stars. Speaking of myelin, we mentioned that before. Where on a neuron do you find myelin? On what part of the neuron would you find myelin? On the axon. Are all neurons myelinated?
No.
No, some are just naturally unmyelinated. But if you have a neuron that has an axon that's myelinated, it's able to do what?
Faster.
Yeah, it can send its action potential a lot faster than ones that are unmyelinated, about 5 times faster. Okay. We then jumped to my favorite slide of all. What's the name for a cluster or a grouping together of neuron cell bodies in the central nervous system? That's a nucleus. What's the name for a bundle of axons in the central nervous system? That's a tract. What's the name of a grouping together or a cluster of neuron cell bodies in the peripheral nervous system? That's a ganglion. Good. And then what's it called with a bundle of axons in the peripheral nervous system? That's a nerve. All right, make sure you know those things. White matter, what makes white matter white?
Myelin.
Myelin. What makes gray matter gray? No myelin, right? So remember, the dendrites, the cell body parts of neurons, they don't have myelin on them. And then remember, there are some axons that are just naturally unmyelinated. If you were looking at the spinal cord on a cross section, where would you find the gray matter? On the spinal cord, the gray matter
It's
is on the inside, and the white matter is on the outside. What about with the brain?
The gray matter
It's
on
pretty
the
much the
outside.
opposite, right? The gray matter is on the outside in the cerebral cortex, and then most of the inside is going to be white matter, unless you go really deep, and then there's more gray matter called the basal nuclei. All right. True or false? Neurons are the only cells that are electrically excitable. False. Remember, muscle fibers are also electrically excitable. We actually covered them first. So we started talking about the electrical signaling in neurons, which is kind of a tricky topic. Before a neuron can even send an electrical signal, though, it has to be ready to do so. And what's that kind of electrical difference called that a neuron has when it's not even doing anything? That's the RMP, or the resting membrane potential. And what was the value for the resting membrane potential? Negative 70 millivolts, which meant that the inside of the neuron was slightly negative compared to the outside of the neuron, right? You've got to have that before you can even do all the electrical signaling that we're going to talk about. Now, when you do the actual electrical signaling, it's usually due to the opening or the closing of different gated ion channels. And there were 4 types of gated ion channels that we talked about. What's the name of the ion channels that like open and close in response to like touch or physical pressure? Those are called mechanically gated ion channels. What's the ones that open and close in response to like a chemical attaching to it? Those are ligand gated ion channels. What's the name of the ones that open and close when the membrane potential changes? Those are voltage gated channels. And then lastly, what are the ones that just randomly open and close? Those are the little leak channels, okay? Resting membrane potential, 3 reasons why the resting membrane potential is negative on the inside compared to the outside. Sodium potassium pump, anions that are stuck inside of the cell and too big to get out, and then lastly, the fact that there are more potassium leak channels than there are sodium leak channels means that more positive ions are constantly leaving and fewer ones are coming in. So all 3 of those factors would set it up so that the inside would be negative compared with the outside. Then we started talking about the actual electrical signaling. We started with graded potentials. They have to happen first. Graded potentials are always caused by some kind of a stimulus. It could be a chemical stimulus, like a neurotransmitter. It could be a touch stimulus. It could be like light rays in the case of your eyes, or sound waves in the case of your. But graded potentials always start with some kind of a stimulus. The stimulus then causes a stimulus-gated ion channel to open, and then you're going to get the graded potential. And graded potentials either look like this, or they look like that. If they look like this, what's that type of a graded potential called? That's a depolarizing graded potential, or it's also known as an excitatory graded potential. Or if you're specifically looking at like the postsynaptic neuron, they have a fancy abbreviation for it. They call it an EPSP, an excitatory postsynaptic potential. So all of those things basically mean the same thing. Whether you call it depolarizing or excitatory or an EPSP, they're referring to this, okay? And those are due to the opening of stimulus-gated what? Do you remember? Is it stimulus-gated sodium channels or stimulus-gated potassium channels? It's stimulus-gated sodium channels, okay? When you open up a stimulus-gated sodium channel, sodium rushes into the neuron and it becomes more positive, right? The other type of graded potential you have is this guy. What's this one called? That's a hyperpolarizing graded potential. It's also, you could also just call it a polarizing graded potential, or again, if you're talking about you could call it an inhibitory graded potential, that's another name for it, or you could call it an IPSP if you're specifically talking about the postsynaptic neuron. So any one of those names, and those are due to the opening of stimulus-gated potassium channels, right? When you open up a stimulus-gated potassium channel, potassium rushes out and the inside becomes more negative, okay? Other stuff to know about graded potentials, the size of a graded potential is based on what? How strong the stimulus is. So if somebody just like lightly touches you on the arm, you might get a graded potential that looks like this. Somebody touches you more forcefully, it might be like that. And then if they really grab you on the arm, it might be like that. So the strength of the stimulus determines how big the graded potential is, okay? The other thing to know about graded potentials, hold on, make sure I get all this here. Remember that one graded potential, hold on a second. Graded potentials have the ability to add together to make bigger graded potentials. What's that property called? Summation, all right? So they can add together. We'll talk more about summation in a little bit, but that's a property that graded potentials have. The question you're always asking with graded potentials is, did you reach what? Threshold. And what's the magic number for the threshold? Negative 55 millivolts, right? So you're starting at negative 70, that's the resting membrane potential, and the question is, did the graded potential cause you to reach negative 50 or not, okay? I mean negative 55, sorry. If you perfectly hit negative 55, what's the result of that? You get one action potential, okay? What if you fall short and you don't quite hit negative 55? No action potential, end of story. And what if you go above and beyond and go over the negative 55?
More than one?
You get more than one action potential, okay? And when an action potential begins, what's the name of the spot on the axon where it's going to start? It's called the trigger zone. That's where the action potential will start, and then it will rapidly move down the length of the axon, okay? Action potentials, they maintain their strength as they go down the length of the axon. They don't die out the way that graded potentials do. So action potentials are used for like long distance communication. Also remember, when you look at an action potential, it sort of looks like this diagram here, okay? That's an action potential. And there's two main phases to an action potential. There's the phase where you're going up like that, and that's called the what phase? That's the depolarizing phase, and that's due to the opening of voltage-gated sodium channels. So sodium rushes in, and you go up like that. And then once you get to the top like this, then you start to open up voltage-gated potassium channels, and potassium will rush out, and so you become negative again. What's that phase called when you become negative again? That's the repolarizing phase. It's bringing you back down to the resting membrane potential, okay? True or false? Action potential, the size of an action potential is based on how strong the stimulus is.
False.
False.
They're all the same size.
They're all the same size. You either have them or you don't, but when you have them, they're always the same size, okay? It's not like a graded potential where you can see different size graded potentials. Action potentials, they're always the same size, okay? Another thing about action potentials that graded potentials don't have is action potentials that have waiting periods where they're not quite ready to send another action potential again. What's the name for those waiting periods? Refractory periods. And there are two types of refractory periods. What's the type called where no matter how hard you try, it's just too soon and the neuron can't do another action potential? That's the absolute refractory period, okay? And it has to do with resetting all those little gated ion channels. It takes a moment to get them reset so that they're ready to open and do their jobs again. So if you try to stimulate a neuron too soon, it's just not going to happen, and that's the absolute refractory period. If you wait a little bit longer, you can do it, but you're going to need to use a stronger stimulus than you ordinarily would. What's that period called? That's the relative refractory period, okay? Stronger stimulus is needed, but you can do it. And then finally, if you wait long enough, then you'll just be back to normal and you can use a regular stimulus again, okay? All right. We mentioned myelination as speeding up the signaling. Specifically, when you have a myelinated axon, the signal is able to jump. The action potential is able to jump ahead from what to what to what? From each node of, what do you guys call it, Ranvier? From each node of Ranvier, you can jump ahead like that, right? What's the name for that when you have conduction where you jump ahead like that? That's saltatory conduction. Like saltatory literally means like jumping. Versus what's the type of conduction you have in an unmyelinated axon called? That's continuous conduction. It's a lot slower, right? Besides myelination, what were two other factors that can influence how fast an action potential is? The size, so the diameter of the axon, and then what else? The temperature. Hotter is faster, slower is colder, okay? Based on myelination and the size and the action potential speed, you can classify axons into three groups. What type of axons send the slowest signals? C fibers, all right? That's because they have a small diameter and they're unmyelinated, so they send it slow. What type of fibers send it the fastest? A fibers. That's because they're myelinated and they have a big diameter and they send it, at about 130 meters per second, which is really fast. Like your touch and your pressure sensations, things that are like coming from those Meissners and those Pacinium, those are usually carried by neurons that have these A-type fibers. Also, a lot of your motor neurons have those A-type fibers. And then in between the two extremes, of course, are the B fibers do stuff. How does your brain know the difference between a weak stimulus and a strong stimulus? Two ways. So the frequency of the action potentials. How many action potentials are coming into your brain over a period of time? Like if you took a 5-second interval, and during that 5-second interval you saw one action potential, your brain would be like, all right, that's not that strong of a stimulus. Versus if you took the same 5-second interval and you had action potential all crammed within that 5-second, your brain would be like, oh my God, that must be a really strong stimulus. So the frequency of the action potentials, and then what's the other thing? Number of neurons that get involved, that get recruited, right? If somebody were to touch you lightly on the arm, maybe only 2 or 3 neurons would get involved, and you're reading like, that's not that strong of a stimulus. Versus somebody like really forcefully grab you on the neuron, maybe 30 or 40 neurons would be stimulated, and they'd be like, wow, that's a strong stimulus. Okay. Types of, we talked about synapses. Synapses are where electrically excitable cells connect. The kind of synapses that we've been talking about are where you have a synaptic cleft, you have a little space, so the first neuron has to release a chemical called a neurotransmitter that crosses that synaptic cleft. What are those types of synapses called? Those are called chemical synapses, right? Because you got to release a chemical to cross it. Versus other types of synapses, the cells are directly attached to each other, so you don't have to release a chemical between the cells, right? They're directly attached. What are those synapses called? Electrical. Which are faster, electrical or chemical? Electrical. Every time you have to cross a synaptic cleft, it slows things down. Okay, so electrical or faster. Let's see here, moving right along. Neurotransmitters. So we were talking about neurotransmitters. After a neuron releases a neurotransmitter, it usually wants it to then signal the postsynaptic neuron, and then it wants it to stop signaling. And in order to stop signaling, you got to get rid of the neurotransmitter. What are the 3 ways that you can get the neurotransmitter out of the synaptic cleft? So you could have an enzyme that breaks it down, right? A good example of that was acetylcholinesterase, breaking down acetylcholine. What's another possibility? Diffusion. So you could just have the neurotransmitter move out of the way, go hang out with a nearby glia or something, just move out of that synaptic cleft. And then what was the 3rd way? Reuptake. Have that first neuron that just released it, have it bring it back in. Okay? It really doesn't matter which one of those 3 you do, just so long as the neurotransmitter is not sitting around in the synaptic cleft. Then we talked about summation. Again, summation is the ability to add together graded potentials to create bigger and bigger graded potentials. You can only do summation for graded potentials because they don't have a refractory period. You can't do summation for action potentials because they have refractory periods. 2 types of summation that you can have. What's this type called, where you have multiple neurons simultaneously stimulating the same postsynaptic neuron? That's spatial summation. So multiple presynaptic neurons all simultaneously stimulating one postsynaptic neuron will allow those graded potentials to be added together and possibly reach threshold. The other type of summation would be where you just have one presynaptic neuron stimulating one postsynaptic neuron, but the presynaptic neuron does it very rapidly. And if it does it fast enough, those graded potentials can add together in the postsynaptic neuron and possibly reach it. What's that type of summation called? That's temporal summation. Okay. Whoops. Classification of neurotransmitters. Why is it tough to classify neurotransmitters based on their function? Just because I said it is? Like, why?
Oh, because they have multiple functions.
Because they might have multiple functions. They might have different, you could have the same neurotransmitter have different functions depending on where it is in the body. In one area of the body, the neurotransmitter might be excitatory, but then in another area of the body, it might be inhibitory. So it's really hard to put it in nice, neat categories if you're basing it on function. Instead, a better way to classify them is just based on their structure. And so if it's big, if it's like a big neurotransmitter, physically big, they call those neuropeptides. That would include things like endorphins, enkephalins, the mysterious substance P, whatever the hell that is. And then there's the small set of neurotransmitters, the small molecule neurotransmitters, like acetylcholine, serotonin, dopamine, epinephrine, all of those guys. Okay. Speaking of neurotransmitters, you can modify neurotransmitter effects in different ways. This is important information if you're interested in ever working for a drug company. When you have a drug or a chemical that's designed to act like a natural chemical in your body, what do you call those types of drugs? When a drug is designed to act like a natural chemical in your body, you call that an agonist, right? When it's designed to block the effects of a natural chemical in your body, what are those type of drugs called? Antagonists, or the slang word that they usually use, they call them blockers. Okay, that's just the slang for them. Neural circuits are when neurons are grouped together into different patterns. Neurons never just sit all by themselves. They're always hanging out with other neurons and they can form different patterns. What's it called? What's the neural circuit called when you have a whole bunch of neurons stimulating a smaller group, and then just eventually one neuron? That's a converging pattern. What's the exact opposite called when you have one neuron stimulating many more? That's diverging. What's it called when you have kind of a combination of the both, where it starts out diverging, but then it comes back and converges? That's a parallel after discharge circuit. And then what's the one called where you have one neuron stimulating a second that then gives off a branch that re-stimulates the first neuron? That's a reverberating circuit. Okay. As you grow and learn new things and experience new stuff, your neurons will make new synapses. They'll grow they can grow branches to their axons or branches to the dendrites, and they'll make new synapses. And that ability for your neurons to change with learning, that's called plasticity. Okay. Neurons are not good at making more of themselves, though. They're not good at mitosis. That's called neurogenesis. 3 reasons why they're not good at neurogenesis. 1, the neuroglia, as great as the neuroglia are, they have an inhibitory effect on the neurons, it seems, especially oligodendrocytes for whatever reason. Number 2, when you're an embryo or a fetus, your neurons can undergo mitosis because you have growth chemicals that promote that. After you're born, though, you don't have those special growth factors anymore, so that's another reason. And then lastly, when you damage your nervous system, scar tissue forms very rapidly, and that can prevent new neurons from growing. That said, there is evidence in certain brain areas that neurons actually can undergo neurogenesis. For example, the hippocampus, they have found evidence that neurons actually can undergo mitosis in that area. But most places in the brain, you don't get it. You're not going to get new neurons. You're not going to get mitosis. In the peripheral nervous system, if you damage a neuron, like if you cut the axon of a neuron in the peripheral nervous system, it can potentially repair itself, but 3 conditions have to be met. The cell body of the neuron has to still be alive, the Schwann cells still have to be functional, and then scar tissue can't form too rapidly. If you can meet those 3 criteria, and you're in the peripheral nervous system, you might be able to repair the neuron. 1 to 2 days after the injury to like if you cut the axon, 1 to 2 days after that happens, the Nissl bodies are going to break up, and that's called chromatolysis. Okay, 3 to 5 days after the injury, the axon beyond the point where it was cut is going to dissolve, and that's called Wallerian degeneration. Then after that, the Schwann cells that are still alive, and actually after that, macrophages will come in and they'll kind of gobble up and clean up the area. And then after that, the Schwann cells will form something called a regeneration tube that will hopefully help the axon to regrow, help guide the growing axon so that it connects back up with like the muscle it was going to, or wherever that particular neuron was going to. All right, got it forever. Any questions about chapter 12? All right, chapter 13 then, spinal cord and spinal nerves. Spinal cord is part of your central nervous system. Spinal nerves are part of the peripheral nervous system. Again, if you do a cross-section of the spinal cord, you find white matter on the outside and gray matter on the inside. The white matter is going to have myelinated axons that are either going up or coming down. If they're going up, what kind of tracks are those? Those are sensory tracks. And if they're going down, they are motor tracks, right? That's in the white matter. In the gray matter, You're gonna have basically a lot of little interneurons and things. That's a place where summation can happen, where you can integrate IPSPs and EPSPs and kind of add them together and see what happens, okay? Anatomically, where does the spinal cord begin?
The brainstem.
Foramen magnum, that big hole, and then anatomically, where does it end? L1, L2 vertebrae. Someone in class this morning said C1, C2, and I was like, that's a super short spinal cord, right? Anyway, L1, L2 is usually where it ends. Remember, helping to protect. What do you have that helps protect the spinal cord? The vertebrae, and then also deep to the vertebrae, you have the meninges. What's the outermost meninge called? Dura mater. What's the middle layer called? Arachnoid mater. And then what's the name of the space under the arachnoid mater? Subarachnoid space. What's in that space? CSF, cerebral spinal fluid. And then what's the name of the layer that's directly attached to the spinal cord? The pia mater. Okay. So those are the three layers. One thing I want to tell you, I don't think it's on the exam on Thursday, but it comes up on the final exam. You're used to hearing the word meninge. I guess technically speaking, the word meninge is like a plural word. There is a singular form of meninge, and this is it, meninx. So don't be, if you see this tomorrow, if you see this on Thursday, or if you see it on the final, it just means the same thing as meninge. It's just kind of a snobby way of saying the singular form of meninge. I didn't write it, but anyway, yeah.
During the lab review, you talked about the 2 layers of the dura mater and how we'll need to know those for the lecture exam.
Yes,
That's this one?
yes.
Okay.
I always say yes to do I need to know questions. Yeah, that's just me. Whether it's true or not, I just always say yeah. It's just how I roll. All right. Anyway, moving right along. Oh, the pia mater. So the pia mater is important. It's the innermost layer. There's going to be blood vessels in there that are giving blood to the spinal cord. Also, the pia mater has some extensions that help anchor the spinal cord in place. What's the name of the extensions of the pia mater that come off the sides that kind of hold it from shifting to the left and to the right? Those are the denticulate ligaments. So they hold the spinal. And then what's the name of the extension of the pia mater that's kind of like a little rope that's at the very end that connects it down to the coccyx?
Horsetail.
Well, it's in the middle of the horse's tail. It's called the filum terminale, okay? Again, it's just all those things. The denticulate ligaments, the filum terminale, they're like anchors that prevent the spinal cord from shifting around too much. Okay? If you look at the spinal cord itself, like if you look at somebody's spinal cord from the back, is it the same thickness all the way down? No. Where is it bigger?
2 spots.
Cervical and the lumbar area. Those are called the cervical enlargement, the lumbar enlargement. Why are they bigger in those areas? There's extra nerves coming in for your extremities, right? So cervical enlargement, extra for the upper extremities. Lumbar enlargement, extra for the lower extremities. All right. Remember the way that the spinal nerves attach to the spinal cord is using things called the roots. And there's a dorsal root and there's a ventral root. How can you tell the dorsal root from the ventral root? There's a bulge on the dorsal root, and there's not one on the ventral root. Why is there a bulge? What's the name of that bulge on the dorsal root? That's the dorsal root ganglion, and inside of it are going to be the cell bodies of what types of neurons? Sensory neurons. So you hear dorsal root, always think sensory, and then if you hear ventral root, always think motor, right? No, we're not done. Hold on a second. Let's see here. Just trying to think what's the next thing I should torch you with. No, that's good. I think we're good there. All right, I'm going to keep moving along here. All right, so spinal nerves, they're part of the peripheral nervous system. You have different layers of connective tissue that help hold the spinal nerves together. What's the layer of connective tissue called that surrounds each individual axon? That's the endoneurium. If you put a bunch of axons together, you get something called a fascicle. What surrounds the fascicle? The perineurium. And then if you put all the fascicles together, you get the entire nerve. And what surrounds the entire nerve? The epineurium, right? Again, that should look very familiar because it was the same kind of setup that you had for muscle, except we use mysium instead of neurium, right? How many pairs of spinal nerves do you have? 31 pairs of spinal nerves. Except for C1, all of the spinal nerves exit through holes on the sides of your vertebrae that are called the intervertebral foramen. Okay? C1 comes out in between your occipital bone and your first cervical vertebrae, so it's not coming out through an intervertebral foramen, but all the other ones do. Remember that the spinal nerves, as soon as they come out of the spinal cord, almost immediately they start giving off branches. And unfortunately, they don't necessarily use the name branch, they'll use the name ramus or rami, right? What's the biggest ramus that you have? The anterior ramus. And that's going to go basically to give sensory and motor to the sides of your body, the front of your body, your entire upper extremity, your entire lower extremity. So it's a much bigger size branch than the posterior ramus, which is just going to your back and doing like the muscles in the skin of your back, okay? Another thing to know about the anterior rami is they like to interconnect with other anterior rami of different levels. And what's that called when you have them interconnecting like that? That's called starts with a P. That's called a plexus, right? And we'll talk about plexuses in a second. So plexuses are formed when anterior rami connect with each other. Some other branches that you have, there's a small meningeal branch that basically goes back into the intervertebral foramen. It supplies the vertebrae, the meninges, the spinal cord blood vessels. And then lastly, there was something called the rami communicantes, which are a way to kind of connect the spinal nerves up with your autonomic nervous system. We'll get way more into that in chapter 15, the autonomic nervous system chapter. So talking about plexuses. Plexuses are ways of interconnecting the anterior rami to organize the neurons that are going to go to different parts of the body. True or false? All anterior rami form plexuses. False. What's the area where they don't form plexuses? Yeah, pretty much the whole thoracic area, right? The thoracic area, specifically T2 through T12, those anterior rami don't form plexuses. Instead, they become what are called the intercostal nerves, and they kind of go straight out to their target skin or muscle, but they don't interconnect with each other. Everywhere else, though, you're going to have a plexus. What's the highest plexus? You have it up in your neck. Cervical plexus. And what's the famous nerve that comes off of the cervical plexus? The phrenic nerve, right? And why is it famous? It makes you breathe, right? It goes to your diaphragm and it literally makes you breathe. What spinal nerves give off branches that are part of the phrenic nerve? C3, C4, C5 keep you alive, right? All right. Then what's the next plexus you have after the cervical plexus? The brachial plexus, and that's the one that's going to organize the nerves that are going to your upper extremity. Then you get a break. There's no plexus in the thoracic area, but then after that you have the lumbar plexus. What's the biggest nerve coming off of the lumbar plexus? Hold on a second. Is that coming off of the lumbar plexus? No, that's coming off of the sacral plexus. What's the biggest nerve that comes off of the femoral? The lumbar. Sorry, it's been a long day. Lumbar plexus. It's the femoral nerve. The femoral nerve is the biggest nerve coming off of the lumbar plexus. After the lumbar plexus, you have the sacral plexus. That's where the famous sciatic nerve comes off of. And then after that, what's the last little tiny plexus you have? The coccygeal plexus, right? We then talked about dermatomes. Dermatomes are areas of the skin that get their sensation from different spinal nerves or possibly from a cranial nerve, right? True or false, all of the spinal nerves have dermatomes. False. C1 does not have a dermatome. True or false, none of the cranial nerves have dermatomes. False. Remember the trigeminal nerve, cranial nerve number 5, wherever it is. The trigeminal, it does the dermatome, that is your face, so sensory to the face, right? All right, we then talked about the tracts. Again, tracts are in the white matter of the spinal cord, and if they go up, what kind of tracts are they? Sensory tracts go up, and then motor tracts go down. Or sometimes they call them, if they go up, they're called ascending tracts, If they go down, they're called descending tracts. Okay. Now, tracts are often named based on where they start and where they end. So the first part of the name tells you where they start, the second part tells you where they end. So, for example, the spinothalamic tract, where does that start? In the spinal cord, and where does it end? In the thalamus specifically, right? So it's starting down low and it's ending up high, so you know it has to be what kind of tract? It's got to be sensory, right? It's going up. Versus like the corticospinal, where does that start? That starts in the cerebral cortex, and where does it end? Spinal cord. So it's starting up high and it's ending down low, so it has to be what kind of tract? It's got to be motor tract, right? In terms of what these tracts do specifically, remember this. Let's go into that. So let's talk about the sensory tract first. I mentioned the spinothalamic tract. What's the spinothalamic tract famous for? What sensation? Pain. That's what it's most famous for. But it also does temperature sensations in your skin, hot and cold. It does itch and tickle sensations, deep pressure, crude touch. All of that stuff is then carried by that. The posterior column tracts, which include the gracile fasciculus, the cuneate fasciculus, they do discriminative touch, light pressure, vibration, and then conscious proprioception. What's conscious proprioception? Kind of knowing where your body parts are in space without having to look, like knowing are your arms out in front of you, are your legs bent, all that kind of stuff. That's conscious proprioception. So those were the sensory tracts. We will talk about, they will raise their ugly heads again in chapter 16. So they do come up again. So they're good. It's good to know these tracts for this next lecture exam, but they also raise their ugly head on the final a couple of times. The motor tracts come in two categories. Some motor tracts are classified as direct pathways and some are classified as indirect pathways. Direct pathways control voluntary movements of your skeletal muscles, whereas the indirect pathways control automatic movements of your skeletal muscles, okay? The direct pathways, they all begin with the prefix cortico because they all start in the cerebral cortex and then they go down from there. And you have things like the corticospinal tracts, they control voluntary movements of your arms, your legs, your trunk. And then you had something called the corticobulbar that starts in the cerebral cortex. The bulbar part's a little bit weird, but that literally means like the brainstem. So they're starting up high, and then they're going down and stopping in the brainstem. It's actually that tract that connects up with your facial nerve and does voluntary movements of your facial muscles. Okay? So those are direct pathways. The indirect pathways, they start a little bit lower in the brain. So they don't start in the cerebral cortex. They start usually in the brainstem, and then they go down from there. And they have a bunch of crazy names based on exactly where they start in the brainstem. For example, there's a rubrospinal tract. Rubro literally means red, and it's called that because it starts in the red nucleus of the midbrain, and then it goes down to the spinal cord. Or the tectospinal tract, for example, that starts in the tectum of your midbrain, and then it goes down to your spinal cord. So all of the indirect pathways, they start lower, someplace in the brainstem, then they go down. And again, they're doing automatic movements. They help maintain posture, regulate skeletal muscle tone, etc. Lastly, we finished off with reflexes. So with reflexes, they're basically like a negative feedback system. You're monitoring some body condition, sending the information in to the central nervous system. It gets analyzed, and if there's a problem, then it sends a signal out to try to fix it. That's essentially the same thing as a negative feedback system. 5 parts to a reflex arc. What's the first part of the reflex arc? The sensory receptor. So that's monitoring some body condition. What's the second part of the reflex arc? The sensory neuron. That's carrying the signal in towards the central nervous system. What's the third part? Yeah, the integrating center, or you could also call it the control center. That's usually going to be someplace in the central nervous system. It's going to analyze the information and decide if anything needs to be done. And then if it does, what's the fourth part of the reflex arc? The motor neuron. That carries the signal away from the central nervous system. And then what's the fifth and last part called? That's the effector. And again, if it's a somatic reflex, the effector is always going to be what? Skeletal muscle. If it's an autonomic reflex, the effector is going to be smooth muscle, cardiac muscle or glands, right? Lots of ways to classify reflexes. What are reflexes called where you have the motor neuron, where you have the sensory neuron coming in and it directly synapses with the motor neuron that's going out? What's that called? That's a monosynaptic reflex because there's just one synapse between the sensory neuron and the motor neuron. What's it called if the sensory neuron comes in, synapses with an interneuron, and then the interneuron synapses with the motor neuron that goes out? That's a polysynaptic reflex, right? You've got more than one synapse, right? Which kind of reflexes are faster, monosynaptic or polysynaptic? Mono, right? Every time you have to have a synapse, it slows things down, so the fewer synapses, the faster the reflex is going to be. Another way to classify reflexes is based on whether they're ipsilateral or contralateral. What does it mean if you're an ipsilateral reflex?
Same side.
It means same side. It means that the side that the sensory signal is coming in is the same side that the motor signal is going out, right? And most of the reflexes we studied were ipsilateral reflexes. So we studied stretch reflexes, Golgi tendon reflexes, flexor withdrawal reflexes. They're all ipsilateral reflexes. What's a contralateral reflex?
Opposite side.
Opposite sides. It means the sensory comes in on, say, the right side, but then the motor goes out on the left side, okay? And there was only one reflex that we studied that was a contralateral. What was that? Don't all answer at once. That was the crossed extensor reflex. Give it up for the crossed extensor reflex. All right. Another thing about reflexes, they all have reciprocal innervation. What does that mean? It just means that when you have a reflex, you're going to be telling muscles to do something, but you've got to remember every joint has multiple muscles crossing it, and if you're telling one group of muscles to do one thing, you better make the opposing group do the opposite or the reflex isn't going to work. So when you do the knee-jerk reflex, you're going to be telling your quad to contract, but at the same time, you also got to tell your hamstring to relax. That's reciprocal innervation, right? You got to make sure that all the muscles are kind of in agreement and that they're working together to make the reflex happen. Okay. All right. Chapter 14, last but not the least. This is the brain and cranial nerves. What division of your nervous system is the brain in?
CNS.
Central. What division are the cranial nerves in?
CNS.
All right, good. Peripheral. That's at least one question you get right. What are the four major areas of the brain? Brainstem, cerebellum, cerebrum, and diencephalon. Those are the four major parts. What are the three subparts of the brainstem? Medulla, pons, midbrain. What are the three subparts of the diencephalon? Thalamus, hypothalamus, and epithalamus. Good. Blood flow to the brain. Does the brain have a good blood supply?
Yes.
Yeah. It has a very high demand for oxygen and glucose. It doesn't store any glucose, so the only way to meet its needs is to have a lot of blood supply. You got two main arteries in the front of your neck, a left and right internal carotid artery that bringing blood up in the front of your neck, and then in the back of your neck, you've got a right and left vertebral artery that are bringing blood up. So those are the arteries. They're bringing the fresh oxygenated blood up to your brain. And then what's the name of the big veins that you have in your brain that collect the used deoxygenated blood? Those are the dural venous sinuses, all right? They collect the used blood, and then they take it back down using your internal jugular veins that are going to bring it back to the heart, okay? Blood-brain barrier. What's the purpose of the blood-brain barrier? To stop things that might be harmful in your blood from being able to cross through the walls of the capillaries and get into your brain tissue where it might damage your brain tissue, right? CSF, cerebral spinal fluid. So let's talk about this. Again, you told me this before, but refresh my memory. What is the name of the glial cells that make CSF? Started with an E. Ependymal cells, right? They make the CSF. And they are located in these little spots called choroid plexuses, which are usually inside of the ventricles of your brain, right? And how do the ependymal cells make CSF? They just do it by filtering your blood, okay? They filter your blood, they take out a lot of the liquid part of your blood, they leave the cells behind, and that liquid becomes the CSF. So you make the CSF by the ependymal cells in the choroid plexuses, which are inside of the ventricles, and let's walk through then the circulation of the CSF. So let's suppose you made some CSF and you're in the lateral ventricles. Where would the CSF go after the lateral ventricles? So through these little passageways called the interverticular foramen, then you wind up where? Then you're inside the third ventricle, which is right in the midline. Where do you go after the third ventricle?
Aqueduct.
Go through the cerebral aqueduct. Then you're in where?
The fourth ventricle.
Fourth ventricle. Once you're in the fourth ventricle, you have options, right? You could continue on inside the central canal, inside the spinal cord. That's one option. The other option, though, is there's some holes in the fourth ventricle called the apertures, the median and lateral apertures. And those holes will allow the CSF to move out and get into where? The subarachnoid space. So that's how the CSF moves from being inside your central nervous system to now being on the outside in that subarachnoid space. Eventually the CSF circulates around the subarachnoid space until it comes to where that returns it back to the blood, the arachnoid villi. Okay, at the arachnoid villi, it will cross back into the blood at that point. So you make CSF by filtering the blood, it circulates, and then ultimately it has to be returned back to the blood. Okay. What's it called if you have a blockage and CSF starts to build up in your brain? That's hydrocephalus, right? All right, we talked about the brainstem first. The lowest part of the brainstem is the medulla. The medulla has a lot of important nuclei in it. Again, the definition of a nuclei is a cluster or a group of neuron cell bodies in the central nervous system. And those nuclei that are in the medulla, those are the guys that control your vital functions. There's a cardiovascular center that controls your heart. There's a medullary rhythmicity center that controls your breathing. There's even centers that control like vomiting and swallowing and sneezing and things like that. So the medulla is a super important part of your brain because of those centers. Likewise, the pons is pretty important too. The pons has some breathing centers in it. There's something called the pneumotaxic and the apneustic areas. They assist the medullary rhythmicity area with breathing. Then above the pons, you get into the midbrain. Probably the most notable feature on the midbrain is on the back of the midbrain. What do you see on the back of the midbrain? The four corpora quadrigemina, right? You see those four bumps. The upper two bumps are called the what? Superior colliculi, and the lower two bumps are the inferior colliculi. Remember, the superior colliculi, they handle visual reflexes. So when you're looking at a scene with your eyes, if something moves in that scene, I don't know, like a little rabbit starts moving around, we have a natural reflex built in where we'll turn our body and our eyes and our head towards that movement. It's sort of a survival mechanism. And, because who knows, that rabbit may have been a lion or something that we want to run the hell away from, right? So we turn and look at it like, oh, good, it's just a rabbit, right? But that built-in reflex, that's covered by the superior colliculi. It responds to visual stimuli. We also have the built-in reflex for sounds. A sudden sound will cause us to turn our body, our heads, and our eyes towards the sudden sound, and that's handled by the inferior colliculi. Okay. Other thing in the midbrain that's pretty important is the substantia nigra. That's a place where neurons release dopamine. It helps control subconscious muscle activities. It's an area that loses neurons with Parkinson's disease. And then finally, another really important area of your brainstem that extends through the medulla, the pons, and the midbrain is the reticular formation. It's a combination of white and gray matter, and it kind of has a net-like appearance to it. At least that's what the people that discovered it say. The most famous thing about the reticular formation is something called the reticular activating system, and it's important for three reasons. What did the reticular activating system do? It helps you, number one, maintain consciousness. Number two, it wakes you up in the morning from sleep. And then number three, it helps you filter out the important information from like the garbage, right? So you can stay focused on things. We then went and talked about the cerebellum. What does the word cerebellum mean? It means little brain. That's because it looks like a smaller version of the cerebrum. The cerebrum's the biggest part of your brain. The cerebellum's the second biggest part. The cerebellum has the folds on it, etc., that make it look kind of like a mini version of the cerebrum. On the back side of the cerebellum, I think most people got this right, is that thing that looks like the worm. What's that called? The vermis. Good. And then I think most people got this right as well. What's the name for the white matter inside of the cerebellum? The arbor vitae, right? The tree of life. What is the function of the cerebellum? What does it help you with? Hand-eye coordination. Okay. It also helps with like posture and balance. It's basically, the way I always like to think about it is it's like a quality control center for your body movements. When you want to do a movement, the idea for that is going to start up higher in the cerebral cortex, but the signal is going to be sent to the cerebellum, just kind of saying, heads up, this is the movement that I'd like to do. And then the signal gets sent down through your spinal cord to the actual muscles, you do the movement, and then signals from the muscles get sent back up to the cerebellum. So now the cerebellum knows two things. It knows what you want it to do, and it knows what you actually did. And if the two match up, that's great. But if they don't, the cerebellum will try to fix it so that your actual movements match up with what your intended movements, what your plan was, right? All right. Next, we went into the diencephalon, made up of the thalamus, the hypothalamus, the epithalamus. The biggest part of the diencephalon is the thalamus. Makes up about 80% of the diencephalon. The thalamus is famous for being a relay center for sensory signals that are traveling to the cerebral cortex. Remember, you don't become consciously aware of any sensation until it reaches the cerebral cortex. But most sensations don't go there directly. They stop first at the thalamus, then they synapse with a second set of neurons that carries the signal up to the cerebral cortex. With the exception of what sensation? Smell. Smell goes straight to the cerebral cortex. It doesn't stop at the thalamus. Okay. All right. Another thing to mention, hypothalamus, that's a big important area for homeostasis. The set point for your body temperature is located there. There's also an eating and drinking center that's located in the hypothalamus to kind of regulate those behaviors. And then the epithalamus is probably most famous for that's where you find the pineal gland. What does the pineal gland make? Melatonin. What does melatonin do, or supposed to do? Make you feel sleepy, right? When are melatonin levels high in your blood? When it's dark, right? That's when the pineal gland starts churning out more melatonin. All right, we then talked about the cerebrum. The cerebrum is the biggest part of your brain. The outer part is gray matter. That's called the cerebral cortex. It's about 2 to 4 millimeters thick. If you look at the cerebral cortex, it's got these big folds on it. What's the fancy name for those folds that you see? Yeah, those are, well, the folds themselves are called gyri or gyrus, right? And then in between the folds, you have grooves. If it's a deep groove, you call it a fissure. If it's a more shallow groove, you call it a sulcus, right? The reason why the cortex forms these folds is because the gray matter develops faster than the white matter, and it causes it to kind of fold on itself. I mentioned fissures and sulci. What's the biggest fissure that divides the cerebrum into a right and left hemisphere? That's the longitudinal fissure. What's the fissure that separates the cerebrum from the cerebellum? That's the transverse fissure. What's the name of the sulcus that separates the frontal lobe from the parietal lobe? That's the central sulcus, okay? Now, if you look deep inside, or deeper inside the cerebrum, then it's going to be white matter, which means myelinated axons. And those axons kind of come in three types of tracks. Association tracks are axons that connect different gyri within the same hemisphere. Commissural tracks are tracks that connect left and right, so they cross sides. And then projection tracks are tracks that either go up or go down, kind of like the ones we already talked about in the spinal cord. If they're going up, they're going to be sensory. If they're going down, they're going to be motor. And then finally, if you go deep enough in the cerebrum, you do encounter more gray matter again, and that's called the basal nuclei. The basal nuclei help initiate and terminate body movement. So when you want to start moving or stop moving, it's the basal nuclei that sort of help with that. Then we spent time talking about the cerebral cortex in excruciating detail. Remember, there are 4 main lobes to the cerebral cortex: the frontal, the parietal, the temporal, and the occipital lobes. Remember that the frontal lobe is dedicated to motor function. The parietal, the temporal, and the occipital are dedicated to sensory and association areas, okay? So specifically, if you look in the parietal lobe, in the post-central gyrus is an area called the primary somatosensory area. That's where all incoming skin signals have to go for you to become consciously aware of them. So touch, temperature, pain, itch, tickle, all those skin sensations, they have to reach the primary somatosensory area in the post-central gyrus in order for you to be consciously aware of them. And of course, as I showed you in lecture, you can map that out and it actually looks like a little person, and they call that the sensory homunculus, right? So that's the primary somatosensory area. You're going to have primary sensory areas for all of your sensations. So there's a primary visual area that's in the occipital lobe, or there's a primary auditory area that's in the temporal lobe, etc. In terms of the motor areas, again, you should think frontal lobe, specifically the precentral gyrus, which is right in front of the central sulcus. That's where the primary motor area is. That's where all of your ideas for voluntary movements begin. They all start in that precentral gyrus in the primary motor area, and then they go downward from there. Just like you could draw a homunculus guy for your sensory area, you could draw a motor homunculus for this area. Another important motor area, in addition to the primary motor area, is something called Broca's area. Why is Broca's area important? It allows you to speak, right? Broca's area goes to the muscles of your vocal cords, the muscles of your tongue, your lips, all that stuff to help you speak clearly. In most people, what side of the brain is Broca's area on? Left side, okay? 97% of people, it's on the left side. And then lastly, there are what are called association areas. Association areas are located usually very close to the sensory area, okay? Association areas give meaning to the sensory areas, right? So when you say touch something, you will become consciously aware of physically touching something when the signal hits the primary somatosensory area. But then almost immediately, the signal will be sent from the primary sensory area to an association area that then attaches meaning to it. So you touch a ball, you're consciously aware that you're touching something when it hits the primary sensory area. But then the signal is sent to the association area, and that's when you say, oh, that's a ball I'm touching. I've touched balls. Sorry, I'm just gonna stop. I'm gonna stop. I'm sorry.
I was waiting for it.
I'm gonna stop. Where is HR? Does anyone know where HR is? All right, I'm gonna just stop here. Sorry, Nicole. I'm gonna really get myself in trouble. All right. Anyway, other stuff to be aware of. Brain waves. So remember, you can put an EEG on someone, an electroencephalogram, and you can measure brain wave activity. Remember, you're pretty much just measuring the activity of neurons that are in the cerebral cortex because that's the layer that's closest to the surface, okay? The normal regular brain waves that you see when someone's awake, those are called alpha waves. If someone's really thinking hard, which I recommend avoiding, but if you have to, if you really have to think hard, then you'll get what are called beta waves, which are a little bit faster than the alpha waves. Slow waves that you can see that look kind of strange are called theta waves. They usually indicate that you're emotionally stressed or something. And then lastly, these slowest waves you have are called delta waves, which are completely normal if you're asleep. But if you see them in someone that's awake, that's usually an indication that they have some kind of brain damage going on. Finally, we finished off with the cranial nerves. Actually, not quite the last topic, but we then talked about the cranial nerves. Make sure you know this table really well, okay? You know, again, I could ask you, you know, the good news for the lecture exam is there's no pictures. You don't have to identify anything, but, you know, you should still pretty much know all the information in this table. You know, definitely, oh, sorry, I forgot. Here's a gift. Oh, this isn't a walk in the park, but
We
this
like
is another
gifts.
gift. I told you that you guys, some of you guys probably know this, but I told you the memory trick for like old on Olympus, Towering Tower. There is also a memory trick to help you memorize the classification of each. Does anyone know that memory trick?
Some say marry for money, but my big brother says big brain. No, but my big brother says brains matter more.
You're close, yeah. There's no 4 in there, or else that would mess it up. But yeah, it looks like this. So, sorry, I can't even write it. Some say marry money, but your brother, who I guess is smart, some say marry money, but my brother says big brains. By the way, on some of the memory tricks you'll see on the internet, they don't use the word brains here. You can imagine what they might use. Big brains matter more.
Oh, wow.
Alright? So, sensory, motor, both, sensory, both, motor. Alright, so, you're welcome. Alright?
That's jam me up your own words.
Know that, you know all of this. Just know all of this, all right? That just know everything. You'll be fine. You'll get 100 if you know everything. Definitely make sure you take special note of the ones that have parasympathetic branches. 3, 7, 9, and 10. Okay, those are the ones that have parasympathetic branches. We're almost done. Hold on. Development of the nervous system. Again, it's ectoderm. Specifically, the ectoderm forms this thing called the neural plate. The neural plate should fold completely and close off and be called a neural tube. If it doesn't, then you get a neural tube defect. Again, what's the vitamin you can take to help reduce the risk? Folic acid or folate, you know, whatever you want to call it. Aging, nothing good happens to your brain as you age. Strokes are called CVAs. Remember, there are ischemic ones that are due to like blockages, and then there's hemorrhagic ones, which are due to like an aneurysm rupturing. You actually have blood like going out through that hole. Hemorrhage, emboli, atherosclerosis, those are all common causes of strokes. An emboli, which I didn't mention, anyone know what an emboli is? I didn't mention this before. An emboli is when you have a clot someplace elsewhere in your body, and a little piece of that clot breaks off, and then it travels, and then it gets stuck someplace smaller, like up in your brain in one of the things. So for example, you could have, like there's a common heart condition called atrial fibrillation, where the atria don't contract really well, they just kind of vibrate. Those people that have atrial fibrillation, they can get clots in their heart. And if a little piece of that clot breaks off, it'll travel up through the heart, up through their neck in the carotid, and get stuck up in one of the cerebral things, and they'll have a stroke from that. That's called an embolic stroke, okay? If you have symptoms and they go away very quickly, that's called a TIA, but still take somebody in. You may save their life because they could have a full-blown stroke later on. All right, any questions about any of that stuff? It's a lot of information, I know. You know, if you have any questions, come to my office hours, or shoot me an email and I'll try to clarify it for you. But just nail the PowerPoints really hard, and you should do well.
My micro professor just set out a reminder